Very good afternoon to all the NEET aspirants. Today we are going to have a very interactive and interesting session that is on the previous year NEET questions of the chapter Molecular Basis of Inheritance. So, all the questions beta from 2020 to 2024 we are going to solve it now. So, if you look into the question which has appeared in the year 2020, it is on the first phase of translation. It is on the first phase of translation. What is the first phase of translation? Of course, amino acylation of the tRNA is the first phase of translation, wherein tRNA is attached with an activated amino acid complex. tRNA is attached with amino acid. tRNA is attached with amino acid, which is called as amino acylation of tRNA. Clear? This is the first phase of translation. Second, name the enzyme that facilitates opening of DNA helix during transcription. During transcription he is saying. So, from a double stranded DNA, if you take from a double stranded DNA, one is from 5 prime to 3 prime, the other is from 3 prime to 5 prime. So, at the promoter region, RNA polymerase which is DNA dependent RNA polymerase. This is what ma? DNA dependent RNA polymerase at promoter region will open the helix. It will open the helix and it will initiate the process of transcription. Clear? So, it is RNA polymerase that is facilitating the opening of DNA helix during transcription. If the same statement he asks during replication. During replication, it is DNA helix that opens the double stranded DNA strand. Then DNA polymerase activity is to synthesize or to polymerize the new DNA strand, DNA ligase, it will attach the Okazaki fragments during DNA replication. Last step of DNA replication to attach the Okazaki fragments, that is mediated by the enzyme DNA helicase. Clear ma? Yeah. Next question if we take, yes, here, which of the following statements are correct? Which is a statement based question, and here he has asked with respect to nitrogen bases. In DNA, what are the 4 nitrogen bases that we can see beta? That is adenine, guanine, cytosine and thymine. Adenine of one DNA strand which is from 5 prime to 3 prime, and thymine of other DNA strand which is of antipolar. The 2 nitrogen bases of these 2 strands are connected by hydrogen bonds. Adenine, thymine connected by 2 hydrogen bonds, guanine and cytosine are connected by 3 hydrogen bonds. This we all know, right? Adenine pairs with thymine by 2 hydrogen bonds, guanine pairs with cytosine by 3 hydrogen bonds. This we all know, right ma? So, here what is the correct statement? Whenever statement based questions come, you have to look into the question properly whether he is asking you the correct statement or incorrect statement. Correct statement if you look into, fourth statement if you see ma, adenine pairs with thymine through 2 hydrogen bonds, 2 bonds. Adenine does not pair with thymine, wrong. Adenine pairs with thymine. Adenine pairs with thymine with 3 hydrogen bonds, wrong. It is guanine that pairs with cytosine with 3 hydrogen bonds. Adenine pairs with thymine with only 1 hydrogen bond, it is wrong. Clear? Next, see, this is a numerical question. If the distance between 2 consecutive base pairs is 0.3 nanometers, between 2 consecutive base pairs, what is the distance? 0.34 nanometers. And the total number of base pairs of double helix in a typical mammalian cell, total number of base pairs how many ma? 6.6 into 10 to the power of 9 base pairs, 10 to the power of 9 base pairs. What is the length of DNA? You have to remember this formula. The length of DNA is equal to total number of base pairs total number of base pairs into distance between two base pairs Total number of base pairs is 6.6 into 10 to the power of 9 base pairs into distance between two base pairs is 0.34 nanometers. 1 nanometer is equal to 10 to the power of minus 9 meters, right now beta. So, here 0.34 into 10 to the power of minus 9 meters, minus 9 and this gets cancelled. If you multiply this, the answer will be 2.2 meters. What will be the answer ma? 2.2 meters DNA. Next, complete the flow chart on the central dogma of molecular biology. Who gave us the central dogma of molecular biology? Watson and Crick. Very important, you have to remember. First is DNA replication. So, A is what ma? A is replication. DNA replication, then after the message is copied to mRNA. So, B is transcription. This process is called as transcription. Then the information is translated to protein. So, C will be translation. The process is translation and D is protein formation. The ultimate goal of DNA is to synthesize a protein. So, here if you look into A is replication, B transcription, C translation and D protein formation. So, here the option 2 is the correct answer. Identify the correct statement. Sixth question, again statement based question. Look into it properly beta. RNA polymerase binds with Rho factor to terminate the process of transcription in bacteria, correct? The coding strand in transcription is copied to an mRNA, wrong. Split gene, this is wrong. Split gene arrangement is the characteristic of prokaryotes, wrong. The coding strand is copied to mRNA, wrong. It is the template strand that is copied to the mRNA. In capping, methyl guanosine is added to 3' and wrong. In capping, methyl guanosine is added to 5' end. Split gene arrangement is seen in eukaryotes and mRNA is copied from the template strand, not from the coding strand. So, here RNA polymerase during transcription, RNA polymerase, if it is binding with sigma factor, it initiates the process of initiation, it initiates the transcription. If RNA polymerase is binding with Rho factor, binds with Rho factor, then it will do the process of termination. Clear ma? Yes. Next, if we look into, what is the role of RNA polymerase 3 in the process of transcription in eukaryotes, he is asking. Listen, so if you consider this as a nucleus, inside nucleus we have nucleoplasm, right now beta, inside nucleus we have nucleoplasm and a nucleolus, and here in nucleolus, RNA polymerase 1 will synthesize rRNA, ribosomal RNA, and in the nucleoplasm RNA polymerase 2 and RNA polymerase 3, in the nucleoplasm, 2 will be synthesizing mRNA and RNA polymerase 3 will be synthesizing tRNA or soluble RNA. Clear? So, this you have to remember, rRNA is synthesized in the nucleus and by RNA polymerase 1. In the nucleoplasm, mRNA and tRNA are synthesized, mRNA from RNA polymerase 2 and tRNA is from RNA polymerase 3. So, now if you look into transcribes the role of RNA polymerase 3, he is asking. RNA polymerase 3 will transcribe tRNA, 5S rRNA and snRNA, option 1 is correct. It is not synthesizing mRNA, it is not just synthesizing snRNA, it transcribes, no, it does not transcribe rRNA, it transcribes tRNA, 5S rRNA and snRNA. Next, again a small numerical question. If adenine is 30%, adenine is 30% of the DNA molecule, what in the complete DNA molecule, in the complete DNA molecule he is saying adenine is of 30%. What will be the percentage of thymine, guanine and cytosine he is asking. If adenine is 30%, adenine binds with thymine, right? Thymine will also be of equal weight, equal amount, okay. How much percentage adenine is there? Thymine is also of equal amount. So, thymine is also 30%. Thymine is also 30%. Next, guanine and cytosine. If these both occupy totally 60%, then the leftover percent is 40%, right? Leftover percent is 40%, where both of them will be of equal amount. So, each will be of 20% and 20%. Understood ma? If adenine is 30%, thymine will also be of equal amount. So, both together it is 60%. Guanine and cytosine together it comprises 40, means each will be of 20%. If you look into the options, adenine is 30%, so thymine is 30, guanine is 20 and cytosine is 20. Option 2 is the correct answer. Next question, if you look into ma, yeah, which of the following RNAs is not required for the synthesis of protein? siRNA, which is silencing RNA, okay. What is that? siRNA, just a second, yeah. Just a second. Yes. So, which of the following RNAs is not required for synthesis of protein? For synthesis of protein, we need all the 3 types of RNAs, that is tRNA, rRNA and mRNA. siRNA, that is silence RNA, is not required for the process of synthesis of proteins, okay. Next, which of the following statements about histones is wrong? Wrong statement he is asking you. The pH of histone is slightly acidic. No, the pH of histone is basic. This is due to the presence of basic amino acids like lysine and arginine. So, histones are rich in lysine and arginine, correct. They carry positive charge chain, correct. And histones are organized to form 8 molecules, right. So, what are those 8 molecules? We have histone octamer with H2A, H2B, H3 and H4. 2 copies of these 4 histone molecules will form a histone octamer of 8 molecules. Clear beta? So, what is the correct option here? Option 1 is the correct answer. Next question, if you take, yeah, statement based question again, and this is on genetic code which has asked in 2021 ma. So, here if you see statement 1, the codon AUG codes for methionine and phenylalanine, wrong. If it is UUU or UUA, that will be for phenylalanine. AUG will have a dual function, wherein at one side it will be coding for methionine, on the other side it acts as start codon. It acts as what ma? Start codon, okay. It plays dual function. Second statement, if you see adenine, so the first statement is wrong. AAA and AAG, both codons code for amino acids lysine. Yes, both of them will code for the amino acid lysine, correct. So, here what will be the answer? Statement 1 is wrong and 2 is correct. So, here the option is 3. Here again in 12th question, which is in 2022, one more numerical question, very much important. This is from Michelson and Stahl's experiment, that is the experimental proof of DNA replication. Which method of DNA replication ma? Semi-conservative method of DNA replication, Michelson and Stahl's experiment, which is an experimental proof. Here he is saying in the question 10 E. coli cells with N15 N15 isotope, 10 E. coli cells with N15 N15 isotope of double stranded DNA are incubated in the medium containing N14 N14. They are incubated in the medium containing N14 N14 nucleotide. After 60 minutes, how many E. coli cells will have DNA totally free from N15? Totally free from N15 means he is asking you about light chains. He is asking you about light chains. So, here we have 3 forms of DNA in this experiment. One with only heavy isotope nitrogen N15 N15, this is heavy, N15 N14 is hybrid, only N14 N14 are light chains. Okay. So, here he is asking you at the end of 60 minutes how many light chains you can see. So, after 60 minutes means in E. coli one generation time is 20 minutes beta. In E. coli one generation time is 20 minutes. In that case, after 60 minutes how many generations will take place? After 60 minutes it will be 3 generations, right. After 3 generations how many light chains you can see. Let us get started. So, N15 in N14 means with respect to semi-conservative method of DNA replication, one template strand and one daughter strand will be formed, right. So, it will be N15 N14 and N15 N14, one template strand and one daughter strand. Next generation of 20 minutes, okay, in again in N14 N14. What you will get, as there are 2 heavy chains, these 2 chains acting as template, you will get 2 hybrid and 2 light chains, 2 hybrid and 2 light. In hybrid what do you have ma? In hybrid you have N15 N14 N15 N14, 2 light chains of N14 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 N14. So, in the third generation again they are in the media of N14 N14 medium, okay, again they are in N14 N14 medium, wherein these 2 will be acting as template, right. Each and every strand will be acting as template for the next generation. So, 2 hybrids are formed, 2 hybrid, and how many light chains beta? 1, 2, 3, 4, 5, 6. 6 light chains are formed. Clear? So, if you take one E. coli cell after 3 generations, that is after 60 minutes, 6 light chains are formed. If you are taking 10 E. coli cells, if it is one E. coli cell after 60 minutes, 6 light chains are formed, okay. The same thing for 10 E. coli cells, it will be after 60 minutes 60 light chains you can see, okay. 60 light chains means 60 strands without even single heavy isotope. So, what will be your answer? Option 2 is the correct answer. Next question, if you look into ma, yeah, in an E. coli strain, I gene gets mutated, and its product cannot bind the inducer molecule. If growth medium is provided with lactose, what will be the outcome? So, here the question is from gene expression Lac-Operon model. If you take a Lac-Operon with 5' promoter region followed by operator, then 3 structural genes Z, Y, A, lastly it is terminator. So, in this case he is saying that lactose is provided to the culture medium, but there is no I gene. Okay, I gene got mutated. So, I gene which is far away from the promoter region, I gene cannot send repressor. I gene got mutated, it cannot send repressor. In this case what happens? So, it got mutated, but previously only I gene got attached, might be attached to the operator. Previously, before I gene getting mutated, repressor is already there to the operator. Okay. Now, in this condition, even if lactose is provided and I gene is not giving you the repressor, in this case repressor will not relieve from the operator region and can bind to the lactose, right. Repressor which is already binded to the operator region, it can't come out and bind to the lactose, right. So, here even though if you are producing lactose, the gene expression will not take place, because previously only repressor has bind to the operator in the continuous gene expression process. Okay. So, here what will be the answer, ma? ZYA genes will be transcribed wrong, ZYA genes will not be translated, correct. RNA polymerase will bind to the promoter region, yes, of course RNA polymerase will bind to the promoter region, only Z gene will be transcribed wrong. So, with respect to this situation, ZYA genes will not be translated because repressor will already be binded to the operator region in the continuous gene expression scenario. Okay. So, here the answer will be 2. Next, if you look into, if genetist uses blind approach for sequencing the whole genome of organism followed by assignment of function to different segments, the methodology adopted by him is called as. If the complete sequence is considered, not just exons or the expressed parts of the DNA, complete sequence is considered, both exon and intron. If the complete sequence is considered, exon and intron, exon, intron, exon, intron, that complete DNA chain is considered for sequencing, means it is sequence annotation method. Okay. If only exon region or expressed parts of the DNA are considered for sequencing whole genome, then that methodology is called as expressed sequence tags. Next, DNA polymorphism forms the basis of, both for genetic mapping and DNA fingerprinting. Next, here the question is, which one of the following is the sequence on the corresponding coding strand? If the sequence corresponding coding strand, in the coding strand what is the sequence he is asking? If the sequence on the mRNA is as follows. So, he has given us the mRNA sequence from 5 prime to 3 prime. mRNA sequence will be complementary to the template strand which is from 3 prime to 5 prime. Template strand or non-coding strand. Okay. So, mRNA will be complementary to template or non-coding strand, and this will be similar to that of coding strand. mRNA sequence will be similar to that of coding strand, just in the place of T over here U will be there. In the place of thymine, uracil will be there in the mRNA, except that everything is same. So, if you look into the correct sequence of mRNA here, from 5 prime to 3 prime, in the place of U, T will be there, beta. So, in the place of U, T here, AUG, so here ATG, because it is he has given us the mRNA strand and asking us the coding strand. Now, CAU, again CAT, okay, CGU, again T. Just check whether it is given in a correct order or not, that is it. C. Yeah, correct only. No, just a second. From 5 prime to 3 prime we have to take. So, at 3 prime here it is C. So, here also at 3 prime it should be C. Option 1 is wrong. Here also at 3 prime this we have to consider. At 3 prime it should be C. This will be wrong. And at 3 prime it should be C. It is also wrong. So, the correct option will be 2. Instead of going for the lengthy process in identifying each and every code, you can go for a shortcut whether at 3 prime end if there is C or not, and at 5 prime end if it is A or not. Like that you can use the shortcut method also better. Once you are identifying that, then go for a cross checking and choose the option. So, here the option will be 2 G, and here also it is T. Yeah, here the option will be 2. Yes. Yeah. Next. Yeah. Next. Unequivocal proof that DNA is the genetic material was proved by Hershey and Chase experiment which they have done on bacteriophage. Okay. This unequivocal proof that DNA is genetic material is proved by Hershey and Chase by bacteriophage experiment. Next question, if you look into, given below are the two statements. Given below are the two statements, wherein statement 1 he is saying that in prokaryotic cells the positively charged DNA. Here itself it is wrong because DNA is never positively charged, it is negatively charged. So, statement 1 is wrong. Statement 2, if you consider, in eukaryotes the negatively charged DNA is wrapped around the positively charged histone proteins. So, yes, this is true. Statement 1 is wrong and 2 is correct. Statement 1 is wrong and 2 is correct. Option 3 is the correct answer. Next question. Given below are two statements wherein first he is saying that RNA mutates at a faster rate. True. RNA mutates at a faster rate. Statement 2, viruses having RNA genome and shorter lifespan will mutate and evolve faster. Both they will mutate and evolve faster because in RNA the sugar, the pentose sugar which is present, the pentose sugar which is present is ribose sugar, wherein at second carbon, that is at second carbon, free hydroxyl group will make RNA molecule more reactive, and that is why it is unstable. That is why it is unstable. Next, if you look into, yeah, here both the statements are true. Option 4. See, here a match the following type of question wherein he has given the structural genes and I gene and asking us for what they code on. Gene A will code for trans-acetylase, so here it is option 2. Y will code for permease. I, repressor protein. I gene will synthesize repressor protein. Z gene will synthesize beta galactosidase. So, here if you see A2, B3, C4 and D1, option 1 is the correct answer. A transcription unit in DNA is defined primarily by 3 regions in DNA, and these are with respect to upstream and downstream. Always do remember the coding strand or mRNA strand at 5 prime end promoter region will be present, which is upstream. Okay, coding strand or mRNA, upstream is the promoter region which is at 5 prime end, and at the downstream 3 prime end of mRNA or coding strand, at downstream, that is at 3 prime end, terminator is present. What is present? Terminator is present, and in between promoter and terminator we have structural genes. we have structural gene. So, promoter, structural gene and terminator, this will be the order. Next, if you see Frederick Griffith has given us the transformation principle, finding out the experiments on Streptococcus pneumonia. Okay, so here it is option 2, Frederick Griffith is option 3, transformation. Then Jacob and Monod, they gave us the Lac-Operon model, gene expression Lac-Operon model. Hargobind Khorana, he gave us the genetic code. Michelson and Stahl has given us the experimental proof for semi-conservation method of DNA, which is option 2. Now, if you look into the options, A is 3, B 4, C 1 and D 2. Option 3 is the correct answer. Next, which of the following statements is correct regarding the process of replication in E. coli? The DNA dependent DNA polymerase in 5' to 3' direction, true. Always do remember, whether it is DNA synthesis or RNA synthesis, it will be taking place from 5' to 3' direction. If you look into the semi-conservative method of DNA, where each strand will act as template, okay. Leading strand, which is from 5' to 3' region, leading strand will be towards the replication fork, lagging strand will be away from replication fork, which is again from 5' to 3'. So, if you look into it carefully, always synthesis is taking place from what strand to what strand beta? From 5' to 3'. Always synthesis is taking place in what polarity? 5' to 3', 5' to 3'. Clear? So, here option 1 is the correct answer. Next, which one is correct product of DNA dependent RNA polymerase? See, look at the enzyme only. If it is DNA dependent RNA polymerase, it is conducting replication or transcription? You tell me. Yes, it is conducting transcription, because RNA polymerase will synthesize RNA strand from 5' to 3', but the synthesis is dependent on DNA. That is why we are calling this enzyme as DNA dependent RNA polymerase. The given template, okay, he is asking us the correct product. Correct product of this enzyme will be RNA strand, which is from 5' to 3' direction, okay. Template is given, so we have to identify a complementary sequence to it, okay beta. So, thymine pairs with adenine, adenine with uracil, because thymine is not there in RNA. Instead of thymine we have uracil, okay. G, A pairs with U, A, C, C, G, A pairs with U, T pairs with A, U, G, G, A pairs with U, A, G, U. So, here it will be from 5' to 3', this will be the correct answer. You just identify it. Yeah, option 3 is the correct answer. A, U, G, U, A, C, C. Yeah, option 4 is the correct answer. Just cross check it once beta, okay. Option 4 is the correct answer. Next, match the following type, wherein RNA polymerase 3 will synthesize what we have discussed in the previous question also, no. RNA polymerase 3 will synthesize snRNA, tRNA and 5S rRNA also. 5S rRNA also. So, here it is option 4. Termination of transcription is with Rho factor. Splicing of exon is done by, splicing of exons not, it is splicing of introns is done by SNRPs, which is option 1. Tata box, it is present at -10 region of promoter, -10 promoter region it is present, which will initiate the process of transcription more easily. So, here what is the answer? A4, B3, C1, D2, option 1 is the correct answer. Next, which of the following is a nucleotide? Nucleotide means it should be a combination of phosphate, sugar and nitrogen base. Phosphate, sugar and nitrogen base. So, uridine means it is a nucleoside. Nucleoside where it is a combination of sugar and nitrogen base, that is uridine. So, here it is a nucleoside, guanine is a nitrogen base, guanosine is again a nucleoside, adenylic acid is a nucleotide, option 2 is the correct answer. Next question, which is recently asked 2024, statement based question, but again focus here. Given below are two statements regarding RNA polymerase in prokaryotes, wherein statement 1, in prokaryotes RNA polymerase is capable of catalyzing the process of elongation. Yes, correct, it is capable of catalyzing the process of initiation, elongation and termination. So, this is correct. RNA polymerase associates transiently with Rho factor to initiate transcription, wrong. When RNA polymerase is attached to Rho factor, it will terminate the process, it will not initiate. If RNA polymerase is attached with sigma factor that initiates the process of transcription. If this is associating with Rho factor, then it terminates the process. So, here what is the correct answer, ma? Statement 1 is true and 2 is false, option 1 is the correct answer. Next question, if we look into, given below are two statements where in eukaryotes there are 3 RNA polymerases in the nucleus in addition to RNA polymerase found in the organelles. Okay. In eukaryotes there are 3 RNA polymerases, he is saying, okay, 3 RNA polymerases in the nucleus in addition to RNA polymerase found in the organelles. True. What are those 3 RNA polymerases? 1, 2 and 3. 1 will synthesize rRNA, 2 mRNA and 3 will synthesize tRNA. All the 3 RNA polymerase in eukaryotic nucleus have different roles, correct. So, here both statement 1 and 2 are correct, option 3 is the correct answer. Next, if you see again match the following question of 2024, histones are positively charged basic protein. So, this is option 3. Nucleosome, DNA wrapped around histone octamer. DNA wrapped around histone octamer. What proteins it has, ma? H2A, H2B, H3, H4. Two units or 2 copies of this. 2 copies of this will form a histone octamer. Okay, like this. Okay. DNA wrapped around this histone octamer is called as what model? Nucleosome model, which is option 4. Euchromatin is loosely packed chromatin, which is active state of chromatin. Heterochromatin is densely packed chromatin, which is option 2. If you look into the options, A is 3, B 4, C 1 and D 2, option 2 is the correct answer. Next, last question, which is in 2024, wherein he has given 2 statements. Statement 1, in the lacoperon, Z gene codes for beta galactosidase, which is primarily responsible for hydrolysis of lactose into glucose and galactose. Of course, lactose, which is a disaccharide, is cleaved to 2 monosaccharides, that is galactose and glucose, by beta galactosidase, This is synthesized by a Z gene. This is synthesized by what, ma? Z gene. So, here statement 1 is correct. Statement 2, in addition to lactose, glucose and galactose can also induce lacoperon model. True. In addition to wrong. in addition to lactose, glucose and galactose can also induce lac-operon model is wrong. Glucose and galactose cannot induce lac-operon model, only lactose can induce lac-operon model of gene expression. So, here statement 1 is correct and 2 is wrong. Statement 1 is true and 2 is wrong. So, option 1 is correct answer. So, yes, this is all about the PYQs of molecular basis of inheritance. In coming up class, we will discuss about another set of PYQs of different chapter. Thank you.